Hello, this is Greg from Gecco. Today, our guest is Professor Julian Kinderlerer, and welcome, Julian, to our interview today on gene editing. If you could, in the beginning, tell us something about yourself, please.
Thank you, Greg. I'm Julian Kinderlerer, former president of the European Group on Ethics in Science and New Technology, emeritus professor of intellectual property law at the University of Cape Town, a former professor of biotechnology law at the University of Sheffield in the United Kingdom, and at the TU Delft, where I was professor of biotechnology and society. I've also acted as a director at the United Nations Environment Programme on assuring that countries had systems in place to allow for genetic modification use in their countries. That's me.
Julian, at the beginning, can you tell us something about gene editing? I mean, this is such a broad topic that we probably need to focus on particular aspects of it. But if you could give a short outline on what gene editing consists of.
Okay, let's start. The human genome, for example, or any organism's genome, is a very long, most linear set of pieces of DNA, which are very, very large. It is now possible to choose to edit and replace, remove, add to that genome in specific places. So let's think about that for a moment. If we want to add something to the genome in a specific place, we have to have some way of identifying where that place is in this long piece of DNA. That's done by having a complementary strand which binds to the DNA, but it has to be specific. How specific can it be when you only have 4 bases which in a particular order define where that binding position would be? It is almost certain that whatever size of recognition sequence you produce, it will recognize more than one position in the genome. The longer it is, the more likely it is to find no other sites to which it can bind, but that isn't very good. So precision is an important issue of where you want to modify the genome. Once you've decided that, there are particular enzymes which will cut the DNA, and they either cut one strand or both strands. They may cut them so that the 2 strands are not quite cut in the same place, or they can be cut at the same place. And that means, again, that you have the possibility of adding, deleting, or various other modifications you can make to the genome, which is then repaired by the cell system itself. So we can do that. Let's think a bit more. Let's take the human being as a starter. I want to replace hemoglobin coding for sickle cell disease, sickle cell hemoglobin S, with the normal one, hemoglobin A. What do you have to do? In the hemoglobin chain, there's 2 chains, alpha and beta normally. And in the hemoglobin chain, beta, the 6th position in that chain is a variant from the one which is found in most human beings. We could replace it in the sickle cell disease with the one that occurs in normal human beings, and that would solve the problem completely. However, how do you do it? The conceptual way to do it is to remove all the cells that are precursors of human red blood cells from that human, modify them in vitro, and then put them back into that individual. Because otherwise, by doing that, you don't have a rejection problem. And now you have a way of producing normal hemoglobin. Think of the cost of that in terms of the fact that the patient is in hospital for a few months while waiting for the whole system to work. The immune system has to be tampered with in order to achieve it. It's extremely expensive, both in financial terms per individual, and also the cost to the individual of not being able to work for a fairly long period of time. Will this work? Only if you've got a lot of money. I'm really concerned that we're selling a concept which won't work, at least in the adult human. It may be possible to modify the embryo at a very early stage in development so that you never have the hemoglobin S expressed, and that may be a way to solve the cost problem. But otherwise, there is no way that this particular technology is going to work. And we have to think about the issues which this imposes on society if we think it's a solution to the many genetic diseases that we have to face. If we turn to plants, it's a totally different story. Okay? With plants, you can regenerate a plant from a single cell. So you can take a leaf, in most instances. You can take a leaf, edit it using the technology that I've identified. It doesn't matter if it isn't quite unique. There are more than one modifications which may occur, because you can now grow up the plants and select the ones which have the modification you wanted, and only those. You don't have to worry about impacting on individual plants. You're now talking about a huge number of plants, and only select those which have been modified in the way you want. So this technology for plants is really, really a solution to the many problems that we might have, where we can add genes, we can remove genes, we can move them around in the genome of the plant, in chromosomes, without concern for multiple impacts, because we can select out those in which the impacts are deleterious. So this technology is very good in that area. We move to animals. Animals have the same problems as humans, not in the sense of taking somebody out of work, having them in hospital, but nevertheless, there are problems with animals because of the long gestation, the long lifetime of most animals, certainly farm animals, and so on. So there are issues with plants which we can ignore. There are major issues which are ethical in terms of the human himself or herself, the finance, the choice of which individual gets it and which individual doesn't get it, whether or not you are increasing the likelihood of other diseases occurring, what are the impacts on the offspring in many generations from that human, none of which apply to plants, all of which apply to humans and animals.
Let's stay maybe at the beginning, first on the general level, what you mentioned. So you said it is incredibly expensive at the moment, but don't you have hope that it will develop, like with all technology, that in the beginning it is incredibly expensive, and the more applications, the more services also, it's getting cheaper by time. And
I don't think so.
don't you have hope that it will be the case here too?
I don't think so. We will always have to take cells out of the human being, of a specific human being. You will have to, or at least kill most of the cells that produce the disease state, then replace them with the modified cells so that the person can now use the modified tissue rather than the tissue which they were producing. That will always be the case. That is expensive. Now, what you can do in theory, and we've got to think about for some diseases, you don't have to remove those cells which are abhorrent. You can simply add the correct ones, and that will result in the human producing the correct form, and so you don't have a disease state. If you take thalassemia is one of the two types of chain in hemoglobin are not produced. So you, instead of forming a tetramer, which is the normal hemoglobin state, you don't form anything except for the chains which just sit on their own, usually. Now, if you can add in the absent chain, you will form hemoglobin. The absence is now repaired without you having to do anything to kill any cells. So in that case, you can do it. But for sickle cell, you can't. So for each disease, you will have different possibilities. And I wanted to stress the problem of what is considered to be the simplest disease to correct, but which will be so expensive for a very large number of people.
So Julian, what would be your conclusion or request to that? To stop financing research in such fields, because gene editing as such will be always too expensive to actually provide it. Let's take sickle cells to the populations in Africa, where financing the healthcare systems is already a problem. So what are the conclusions or requests following?
I think, one, you've got to think about the disease itself. Can you replace without removal of the cells which don't work? If you can, then this technology may well work for a large number of people. In many diseases which are genetically based, there are very few individuals who carry that disease. Should we logically spend the money on those individuals, let's say it was 1,000 or 2,000 in the whole world's population, we can actually probably afford to correct the disease in those people. But think about their offspring. Think about the cost to the society as a whole if you're spending a lot of money on those people relative to everybody else. Should we be doing it? Particularly, you've chosen Africa as an example, particularly in Africa, where quality of healthcare is not necessarily very good, and there isn't enough money to treat everybody. So can you justify solving a problem for a very small group of people, which would then impact on solving other problems for the majority of people? In the case of sickle cell, there are a huge number of people in, say, Nigeria who carry that trait. So that doesn't apply. But some of the muscular dystrophies, there are very, very few people involved. Should we spend the money on them? That's an ethical question. It's a very difficult ethical question.
In this field, especially when it is about gene editing in humans, in theory and practice, when it comes to germline intervention, so actually having an effect also for the generations to come, it is a very specific ethical issue. What do you think about that? Is that a way that the existing taboo at the moment it is shall be maintained, or do you see any ways to say it can be ethical in particular ways, or generally this taboo is not really a good one?
It's a very interesting question. I'll take one example, actually, in my family. My wife and my grandson, therefore my daughter, carry one of the genes for Tay-Sachs disease. If an individual is born with both copies which result in Tay-Sachs disease, that person will get the disease, and it's an awful disease, and the children resulting from any union will die very quickly and very painfully. What we do at the moment is we screen, and if a couple both carry the allele for Tay-Sachs, they're advised not to marry, or at least to get genetic counseling if and when they get pregnant. That's cheaper than removing the allele for Tay-Sachs or modifying it. Okay? Which should we do? It's a very difficult one. My grandson may want to marry a woman, and he finds that she's also a carrier of Tay-Sachs. What's he do? in the Orthodox Jewish communities, especially around New York, the advice is not to marry. Okay? Which is actually very interesting because that results in the trait continuing in the population and increasing in the population. It may be better to advise them to marry and only allow people who both carry the alleles to marry, and then do genetic testing to ensure that their children don't carry both alleles. It'd be a very interesting ethical question to discuss.
Julian, what do you think? Is it really the task of the lawmakers to regulate that, or would you plead more for the individual shall actually decide going this way, or the family, or what are the frames you think would fitting best in the ethics of that?
I think the medical systems should allow for testing, and if the testing shows predilection for a particular disease state, the individual should be counseled, systems should be explained, and then they should make the decisions. But the health systems must provide for testing. For example, the British National Health Service has just agreed to provide testing for the BRCA1 and BRCA2 genes to all individuals who might carry the alleles which are implicated in breast and ovarian cancer. And that makes sense, okay? It's the first that they actually announced that this week. And that makes sense in order to ensure that people know where they stand rather than find out when it's too late.
Let's open a short side branch, because you mentioned the BRCA1 and 2. So two genes actually that were for quite a while patent protected by a company called Myriad Genetics from Utah in the United States. They lost the patents. When it is about such crucial issues, what do you think about patenting genes? What is now actually a question decided for the time being, but it's still a controversy if or if not human genes shall be patentable.
Well, it's much more complex than that. Myriad lost the patent in the United States. America did not lose the patent in Europe. Okay, it still exists. The reasoning behind the patent being invalidated was the broad claim that Myriad had made to all probes for these two alleles. Well, it's a huge number of alleles which are implicated, a number of variations of the particular gene. What they'd done is to claim a very broad right to test for these two genes. And when new technologies came out, which allowed for the testing to be much simpler than the originally envisaged, they claimed that their patent still covered that, and that was the argument. So it's a very difficult one, but should genes be able to be patented? I don't like it, but yes.
One could also say that actually Myriad actually made it a topic. We all remember the old times when breast cancer was a taboo topic. And actually Myriad entering the markets were very, very frank in actually announcing that the test possibilities and the things. So one also could say that part of the job Myriad did was actually to pull out breast cancer and ovarian cancer out of the shade and into the broad daylight, to make it a topic, to say, here we have a test for you to provide, so use it.
Yeah, I agree. That's why I said I think genes, certain aspects of genes should be patentable. That's exactly the point. But the claim should not extend to techniques which are developed after the patent is agreed. It's a very difficult one to define. But when they took out the patent, there was only one test. It was quite a difficult test. 10 years later, it was a very simple test, but their claims still covered it. Okay, that's the problem that may well arise. There are many variants, not just one, of alleles for BRCA1 and BRCA2, which are implicated. I won't say may, they are implicated in a variety of cancers, not just one, not just women. And so we have to be clear, yes, their test helped a lot of people, but in fact, in the end, they were stopping people getting tested because of the cost. So they started providing help, but in the end, they were inhibiting help because the cost was very high.
Let's talk now about gene editing in plants. And actually, we're talking here about CRISPR. So the number one technology at the moment when it comes to there. And we see a change in EU policy towards CRISPR-Cas. And what is it about? So is it gene modifying? Is it not gene modifying? It is not really clear what the standpoint of the EU is and was. So how do you evaluate the policy of gene editing in plants in the EU?
A mess. We had gene modification, which involved the insertion, usually, of genes into the plant using a variety of techniques. We could not specify where in the plant the gene went. It was either by ballistics, which literally means firing a gun at a leaf, or by the insertion using Agrobacterium tumefaciens plasmids. But in either case, it was not clear where the gene would go, in what manner other areas of the plant's physiology might be impacted by the insertion of a new gene. That concern about impact on physiology was possibly misplaced for the same argument I made earlier, that you can produce thousands of plants and select only the ones which appear, at least on the surface, to have the modification you actually wanted without impacting on the rest of the physiology of the plant. Nevertheless, gene editing has a precision which allows you to modify the plant in 1 or 2 or 3 places much more safely, much clearer where the change has happened. With prime editing, which is slightly different, absolutely clarity at where and what you've done. The EU started off concerned that the modification may result in changes which you could not predict and could not see. Now we can say that's not a likely scenario, using gene editing rather than gene modification. So the EU is saying we can now modify our stance and say gene editing is okay. And that is what has been put to the Parliament, which has agreed it, and now each of the countries has to agree in the Council. Many countries are still opposed. They are still scared that changing plants at the molecular level is not acceptable. Now, it's fascinating because plant geneticists have been using ultraviolet light and chemicals to cause mutations in plants for the last 100 years. What those do to the plant, we don't know. How many changes they cause in the plant, we don't know. But those are natural, as described, and therefore don't come under regulatory requirements. It's silly.
Julian, do you think that the EU focuses too much on risks, usually, of new technology? One could have the impression that the EU is always very, very cautious when it comes to new technologies. Or is it that actually, as the EU, the EU must be very cautious when it comes about new technology? That is their duty and an obligation to say that
It's
you
fascinating.
take care?
This is a very fascinating question. The EU has something called the precautionary principle written into its actually treaties. We have to be precaution when changes might be permanent and which might not be good for the society as a whole. The US has exactly the same, they don't call it that, but it has exactly the same for medicines, but not for anything else. They don't call it precaution, but it's there for medicines, and in fact saves a lot of problems for the number of medical treatments like thalidomide many years ago. For foods and plants, the US system doesn't check until after companies have placed things on the market virtually, which is better. They both have their good sides and their bad sides. What happened with Europe is that green organizations, for a variety of reasons, decided that genetic modification should not be used. Now, I would probably argue that most of the arguments they made were on the basis of how do we earn more money for our company or organization or our organic colleagues, rather than a scientific evaluation of what was happening. And that is very serious. But they won the argument in France, in Germany, in Greece, and many other countries in the EU, so that the EU has lost out on the use of genetic modification compared to much of the rest of the world. And the impact on Africa, which didn't have enough food in the first place, has been major. Whereas Europe, there was plenty of food, so it wasn't a particular worry to European citizens. For Africans who took the European position and thought it was scientifically valid, may well have been a serious error.
At the end of our talk, I would like to ask you for prophecy. So if you think about the future of gene editing, what is it what you see? Will the consumers still be, especially in Europe, very critical towards GM? In medicine, do you see that it could be a revolution, or will it really fail at the cost factor?
Direct gene editing in humans, I think, will fail. Indirect, by that is using the technology to make medicines, to make foods, which you can do either in animals or plants or in factories, will revolutionize the production of medicines, revolutionize the production of food additives and food ingredients, because we can make things in fermenters which currently have to grow in the field, for example. What is fascinating is that Europe never rejected the use of gene modification in industrial contexts. So the manufacture, for example, of vitamin C, which is currently primarily a genetic modification process, was never questioned by European consumers.
Julian, thank you very much for this interesting talk. Fellows, this was Professor Julian Kinderlerer being my guest today. See you next time with a new and another interview partner. Gecco, turning challenges into opportunities.